The overall goal of this procedure is to generate lung tissue in culture that is morphologically and physiologically comparable to native lung tissue. This is accomplished by first harvesting the heart and lungs from an adult rat. The next step is to cannulate the pulmonary artery and the trachea and to connect the cannulated lungs to a bioreactor for decellularization. After decellularization and rinsing, the lung is transferred to a sterile culture bioreactor and prepared for seeding. The final step of the procedure is to seed the desired compartment of the lung with a prepared cell population. Ultimately, results can be obtained that show the decellularized extracellular matrix efficiently repopulated with cells that express regionally specific lung markers through immunofluorescence microscopy.
The main advantage of this technique over existing methods of culturing lung cells in vitro is that the cells maintain differentiated phenotypes for an extended period of time, so they can be studied for several weeks in a three-dimensional biologically derived substrate.
For decellularization, the lungs are connected to the bioreactor cap via the vascular cannula, and the cap is connected to the decellularization apparatus. The glass bottle above the lungs is held in place by a ring stand and attached clamp. At this point, cellular material can be removed to produce an extracellular matrix scaffold which can be repopulated with cellular material. The assembly of the bioreactor for seeding and culture of engineered lung tissue is shown in this diagram. The Luer lock connector of the tracheal cannula